tissues.
All living organisms are made up of cells. We define cell as the basic structural and functional unit of a living organism. On the basis of number of cells, organisms are classified into two types: unicellular organism, multicellular organism. Unicellular organisms. Unicellular organisms are made up of single cell. In such organisms, all functions are carried out by this single cell. Example, amoeba, chlorella. In amoeba, a single cell carries out movement, intake of food, respiration, and excretion. Multicellular organisms. Multicellular organisms are made up of many cells. In such organisms, various functions are carried out by a variety of cells. Example, in human beings, muscle cells contract and relax to cause movement. Nerve cells carry messages. Red blood cells transport oxygen. Xylem transports water and minerals, and phloem transports food in plants. Hence, multicellular organisms are complex in nature. In living organisms, each specialized function is taken up by a group of cells at a definite place in the body. Such a group of cells, which is similar in structure and work together, is called a tissue. Example, blood, muscles, xylem, phloem, and cambium are different kinds of tissues. In multicellular organisms, different types of cells perform different functions. This type of distribution of functions is called division of labor. Thus, a group of cells having same origin and structure and performing a particular function is called a tissue. Can you tell the difference between plants and animals? Do they have the same structure and function? Plants and animals are differentiated on the basis of various characteristics. Functional and structural differences. Plants and animals show structural differences as well as functional differences. Plants are immobile. They prepare their food by themselves. They are autotrophic in nature. Animals are mobile. They have to move around in search of food, mates, and shelter. They cannot prepare their food on their own. In other words, they are heterotrophic in nature. Growth of plants is indefinite. It is restricted to tips of stem and roots. Some young tissues in plants divide throughout their life. These tissues are localized in certain regions. Based on the dividing capacity, plant tissues are classified as A. Growing or meristematic tissue, and B. Permanent tissue. The cell wall in plants is made up of cellulose. The growth in animals is limited. Cell growth in animals is more uniform. There is no demarcation of dividing and non-dividing regions in animals. There is no cell wall. The structural organization of organs and organ systems is more localized and specialized in complex animals than in complex plants. Due to this difference, the animals and plants get modified accordingly and adapt themselves to survive in the environment. This modification includes feeding methods of plants and animals. Plants are adapted for a sedentary existence and animals are adapted to active locomotion. This adaptation contributes to the changes in the organ system design of an organism. Plants as organisms are quite impressive in consideration of their diversity and specialized structure. Plants are made up of a number of tissues that work together. Similarly, as multicellular organisms, most plants require vascular tissues for transport to move water and nutrients from one plant part to another. All tissues responsible for growth are located at the tips of root and stem. Depending upon the dividing capacity of the cells, plant tissues can be classified as meristematic tissue and permanent tissue. Meristematic tissue. Let us perform an activity. Take 2 glass jars A and B and fill them with water. Now, take 2 onion bulbs and place one on each jar with its roots immersed into it. Measure the length of roots on day 1, 2 and 3. On day 4, cut the root tips of the onion bulb in jar B by about 1 centimeter. After this, observe the growth of roots in both the jars and measure their lengths each day for 5 more days. Which of the 2 onions has longer roots? Why? Roots of jar A are longer since they have dividing cells at the root tip. Do the roots continue growing even after we have removed their tips? In jar B, there will be no growth of cut roots since the dividing cells are absent. Meristematic tissue performs the function of forming new cells in the plant. The cells of this tissue divide constantly. That is why this tissue is called meristematic tissue or meristem. Meristematic tissues are a group of cells which is exclusively responsible for cell division, thereby producing new cells. These cells are thin-walled, made up of cellulose. The cells are small and protoplasm fills the cell completely with a prominent nuclei. Meristematic cells may be spherical, oval, or polygonal in shape. The cells are compactly arranged and do not have intercellular spaces. Contains dense or abundant cytoplasm and a single large prominent nucleus. Vacuoles are small or absent. All the cells of this tissue are very active. They are found in certain specific regions of the plants that grow. Meristematic tissue is located at the growing points of roots and stems, base of node, base of internode, or at the base of the leaf. According to their position in plants, the meristems are divided into apical meristem, lateral meristem, and intercalary meristem. Apical meristem. The meristems which are located at the growing points of stem, root, and branches are called apical meristem. They enable the root and stem to grow. Lateral meristem or cambium. The meristems are present laterally or on the sides of roots, stem, and are parallel to longitudinal axis. They are found in vascular bundles of dicot roots and stems. The girth and width of the stem or root increases due to lateral meristem. intercalary meristem. These cells have dense cytoplasm, thin cellulose walls and prominent nuclei. Intercalary meristem are located at internodes or stem regions between the places at which leaves attach and leaf bases, especially of certain monocotyledons, example maize. They are active and continuously form a number of new cells. Permanent tissue. The cells formed by the division of the meristematic tissue take up a specific role and lose the ability to divide. As a result, they form a permanent tissue. The process of taking up a permanent shape, size and a function is called cellular differentiation. Cells of meristematic tissue differentiate to form different types of permanent tissue. We all know that plant body consists of variety of cells. Let us demonstrate this by a simple activity. Take a piece of stem of monocot or dicot, example potato, carrot, radish, maize. Cut it into very thin slices or sections. Stain the slices with safranin. Keep it on a slide and put a drop of glycerine. Cover with a cover slip. Observe the slide under the microscope. Observe the various types of cells and their arrangement. Are all cells similar in structure? How many types of cells can be seen? You will find different types of cells in the section of the stem taken. Some of them have dissimilar structure. This demonstrates that plant body has variety of cells. Permanent tissues are subdivided into two groups. 1. Simple tissues. 2. Complex tissues. Simple permanent tissues. These permanent tissues are called simple because they are made up of one type of cells which have common origin and function. They are grouped under categories parenchyma, chlorenchyma, collenchyma, arenchyma, sclerenchyma and surface tissue. Parenchyma. It consists of relatively unspecialized living cells. They are spherical or oval in shape. They are usually loosely packed and have intercellular spaces between them. Few layers of these cells form the basic packing tissue. This tissue provides support and maintains shape of the plant body. The main function of parenchymatous tissue is assimilation and storage of reserve food materials like starch in potato, beetroot, etc. Chlorenchyma. It is present in leaves mainly. Parenchyma contains chlorophyll and performs photosynthesis, and so it is called chlorenchyma. Arenchyma. It is present in aquatic plants such as hydrophytes. Large air cavities are present in parenchyma to give buoyancy to the plants to help them float. Such a parenchyma type is called arenchyma. The parenchyma of stems and roots also stores nutrients and water. Collenchyma. The cells of this tissue are living, elongated and irregularly thickened at the corners with cellulose. This tissue is located in the leaf stalks below the epidermis. It provides mechanical support and elasticity in young dicotyledonous plants. It allows easy bending in various parts of plants without actually breaking it. Sclerenchyma. The cells of this tissue are dead. They are elongated and narrow. The cells are thickened due to the deposition of lignin. It acts as cement and hardens the walls. The walls are so thick that the cells have reduced lumen. The tissue is located in the stems around the vascular bundle, in the veins of the leaves, and in the hard covering of seed and nuts. It provides strength and hardness to the parts of the plant. Surface tissue or epidermis. Activity. Take a freshly plucked leaf of Rio or Tradescantia, Ficus elastica or any fleshy leaf. Stretch and break it by applying pressure. While breaking, keep it stretched gently so that some peel or skin projects out from the cut. Remove this peel and put it in a petri dish filled with water. Add a few drops of safranin. Wait for a couple of minutes and then transfer it onto a slide. Gently keep the cover slip over it and observe under microscope. What you observe is the outermost layer of cells called epidermis or surface tissue. Most epidermal cells are relatively flat. There are small pores scattered in the epidermis of the leaf. Stomata are enclosed by two kidney-shaped cells called guard cells. They control the opening and closing of the stomata. Epidermis is composed of a single layer of living cells, although there are exceptions. Often their outer and side walls are thicker than the inner wall. The cells form a continuous sheet without intercellular spaces. The epidermis is the outermost cellular layer which covers the whole plant structure. That is, it covers roots, stems, leaves, flowers and fruit. The entire surface of the plant consists of single layer of cell called epidermis or surface tissue. The epidermal cells protect the underlying cells. It helps in protection against mechanical injury and invasion of parasitic fungi. The waxy cuticle prevents the loss of moisture from the leaves and stems, example cactus. The stomata of leaves and stems allow gaseous exchange to take place, which is necessary for photosynthesis and respiration. Water vapor may be given off through the stomata during transpiration. Cork. As woody plants grow older, protective surface tissue, that is epidermis, undergoes certain changes. This forms the several layer thick cork or the bark of the tree. Cells of cork are dead and are compactly arranged without intercellular spaces. They also have a chemical called suberin on their walls that makes them impermeable to gases and water. Complex permanent tissues. Another type of permanent tissue is complex tissue, which is specialized for the conduction of water and mineral substances in the plant body. Complex tissues are made of more than one type of cells. All these cells coordinate to perform a common function. Vascular or conductive tissue is a distinctive feature of the complex plants which has made possible their survival in the terrestrial environment. Xylem and phloem are the two types of complex tissues. They are both conducting tissues and constitute a vascular bundle. It is present in stem, roots and leaves. The complex permanent tissues are classified as A. Xylem, B. Phloem. Xylem. Xylem is a complex permanent tissue with thick-walled cells. Most of these cells are dead. Xylem consists of tracheids, xylem vessels, xylem parenchyma and xylem fibers. Tracheids. These are long, narrow cells with walls that are impermeable to water. When tracheids mature, they die and their cytoplasm disintegrates. This leaves a network of hollow connected cells through which water can pass. Xylem vessels. These are tubular structures and are much wider than tracheids. They are arranged end to end and on top of one another like a pipeline. The cell walls at both the ends degenerate when the cells die. As a result, stacked vessels become continuous tubes. This allows free flow of water and minerals in vertical direction. Tracheids and xylem vessels are essential elements since they are directly involved in the translocation of water and mineral substance. Xylem parenchyma stores the food and helps in the sideways conduction of water. Xylem fiber provides mechanical strength to the plant. Phloem. Phloem is a complex permanent tissue which is specialized for the conduction of food and other organic substances from leaves to storage organs and growing parts of plant body. In phloem, materials can move in both directions, which is not the case in xylem. Phloem consists of 4 types of cells, namely sieve tubes, companion cells, phloem fibers and phloem parenchyma. Sieve tubes. They are tubular cells with perforated walls and arranged end to end. The end walls of the sieve tubes have perforated walls. Materials move through perforated walls from one adjacent cell to another. As the sieve tubes mature, they lose the nuclei and most of the other organelles in cytoplasm. The remaining cell organelles stick to the inside of the cell wall. The rest of the cytoplasm can move from cell to cell to carry substances. Companion cells are phloem cells that surround sieve tube elements. Companion cells are living and keep their nuclei and other organelles throughout their lifetime. Companion cells help to control the activity of sieve tube elements. The sieve tubes and companion cells are directly involved in the translocation of the organic substances. Hence, they are commonly described as essential elements. Phloem fibers. They are elongated tapering cells which are found particularly in the stems. The cell walls are thickened. It gives mechanical strength to the plant. Phloem fibers are dead cells. Phloem parenchyma. These cells are alive and filled with cytoplasm. The cell walls are thin. These cells form the packing tissue between all the other types of cells. Phloem parenchyma stores compounds such as starch. Phloem transports food, that is sugars and amino acids, from leaves to other non-green parts of the plants, such as growing stems and roots.